right, I think we can go ahead and get started. So thank you everyone for joining our NK cell therapy workshop today. I'm really excited to have Dr. Shiyu Wang here with us, who is an R&D director of HebeCell Corp. Before I pass it off to Dr. Wang, I just wanted to share a couple company slides to introduce Proteintech to you. So Proteintech offers reagents for life science research. So we offer antibodies and immunoassays. We have a really expansive portfolio of antibodies that encompass over 13,000 targets and cover many different research areas. We also have immunoassays such as ELISA kits and IHC kits, and we have other kits as well, such as antibody labeling kits and cell separation kits. Within our Proteintech brand, we also have our humankind branded cytokines and growth factors. And all our proteins are raised in HEC293 cells so that you can be confident that they have native folding, post-translational modifications, and glycosylation. We also offer both RUO and GMP grade proteins. And I'll talk a little bit more at the end about kind of the advantages of our humankind proteins for NK cell cultures. We also have our Chromatech branded nanobody-based reagents, and nanobodies are 10 times smaller than traditional antibodies, and they can bind to the protein of interest with really high affinity. So they're great for things like clean immunoprecipitation, super resolution microscopy, and protein purification. And then lastly, we also have a subsidiary of Proteintech known as Proteintech Genomics. And Proteintech Genomics manufactures antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. We also value that we are a company for scientists by scientists. So our award-winning products are self-manufactured, and we offer the largest in-house portfolio of antibodies for the human proteome. We also know the importance of product validation and use knockout, knockdown models for validation whenever possible. And as antibody experts, we have in-depth knowledge of immunoassay applications and can offer outstanding technical support. So with that, I want to go ahead and introduce our speaker for today, Dr. Shiyu Wang. So Dr. Wang is the R&D director of HebeCell Corp, an innovative stem cell biotech company. Founded in 2016, HebeCell has developed and patented a method of producing allogeneic natural killer cells from pluripotent stem cells and is now on the track of its first IND filing. Dr. Wang joined HebeCell in 2019 as a scientist after her PhD graduation from Brandeis University. Over the years, her role has gradually shifted from R&D into process development and chemistry, manufacturing, and controls as the company advances towards clinical stages. So with that, I'll go ahead and pass it off to Dr. Wang. Thank you so much for being here today and for sharing your knowledge with us.
Thank you so much for having me. Let me also quickly sharing my slides. Thank you so much for having me, Freda. It's my pleasure to have this opportunity to talk a little bit about what I do, what HebeCell does, in regarding process development for pluripotent stem cell derived NK cell therapy right now. So just like Freda has mentioned, HebeCell is a relatively new company. We were founded back in 2016 and in Natick, Massachusetts. We really started as a very research-based company. Started with things like 2021, so 3 years ago. We experienced the move of this NK cell therapy field moving more and more towards process development and then eventually CMC products. So we started also learning more about process development and then learning more about CMC. Now we have established our own manufacturing process for our first natural killer cells derived from pluripotent stem cells. we have set up all our process development assays, and then we're looking to file R&D like latest, like not latest, like either the end of this year or early next year. Before I go too much into the talk, Mike, I'm a I'm a very interactive like speaker, so I would love to have more some interaction with you all, even through the webinar, if that's possible. So to start with, can you raise your hand if you are still like at school, like master students, PhD students, postdoc in academia? If do you mind raise your hand if possible? Oh, great. Thank you. Thank you. Cool. Thank you. So we have about like maybe like 1/3 of the populations are still at school. Then can you raise your hand if you are in the research and development like department? Cool. 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 Thank you so much for participating. Okay, then like can you now, like if you are in the process development field, do you mind raise your hand? Cool. Cool. Yeah, so this is actually very exciting to see, like we have a lot of people interested in process development, and then like interested like to learn more about process development. So for me in particular, I I feel like I'm still also learning too, like learning more and more about process development, and then I'm sure there are people in in the crowd right now has actually more experience and then more knowledge than I do. So I just hope to share my experience at a very unique situation, which is HebeCell is a very small company. We have like 30 people in total, like including the R&D team, the process development team, the CMC team. We're a very, 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 very small startup company. And then we have this very new technology, and then we have problem finding out a bigger team who can more establish the team or establish manufacturing method that works directly for us. So we needed to figure out how to do the tech transfer, like learning from what we have established in the R&D, and then make sure we appropriately transfer into the process development department and then into this next CMC product. So hopefully I can share a bit more about that, and then hopefully we can have some discussion too. Okay. Let's start. Thank you so much. Before I start talking more about the process development, I want to explain a little bit about the product we have and then the platform we have to produce that product. So at HebeCell, we have a patented, very unique like NK cell platform where we use pluripotent stem cell, and then we culture them, expand them, and then differentiate them into natural killer cell in bioreactors. So the whole process is mimicking our developmental process in our own body from an embryo, how we go from with one single cell and then like differentiate into multiple different layers and then multiple different like specific cell group over time. And this is a picture about what our final NK cell product looks like. They have elongated, like irregular, like morphology, which is very unique and very distinct. When we have our NK cell product, like of course we want to make sure they're actually NK cells. So we look at the receptor expression profile on our NK cell surface, and we found the expected inhibitory receptors, expected activating receptors, like really matching like what our definition of NK cells like right now. Our NK cells are also like cytotoxic. This is an example video about like when we put our NK cells and then osteosarcoma like GFP cancer line together. We can see the osteosarcoma like cancer cells like transfected with GFP, so they're green, and then the NK cells are like not labeled. So you can kind of see this little the shadow of those NK cells moving like across NK cell, like cancer cells, and killing them. They're quite cool. And this is another like example video. Like now we're looking at a phase contrast video. We can see both the cancer cells and the NK cells. The cancer cell patches is like at the center, you can see the big patch, and then this irregular shaped NK cells like getting closer to the cancer cells, targeting them, and then making the cancer cell patch shrink. Cool. Now we have an NK cell product, and then we think is potentially can be a product. How do we do process development? And in this webinar, I'm going to very briefly touch upon like what are the general like guidelines about process development for cell therapy. I'm going to talk a little bit about more specific challenges for pluripotent stem cell derived NK cells, and then talk a little bit about the specific challenges like I or like HebeCell experienced as a very compact biotech company when developing the whole process. To start with, for cell therapy, like process development, there are 4 major criteria that you really want to make sure you have and you understand, and then have release assays that reflect those, which are identity, potency, purity, and safety. For purity and safety, they are more generally applied to all cell therapy. We need to make sure there's like safety means that you should not have bacteria, fungi, mycoplasma, or like viruses or endotoxin in your product. And then you should have very efficient methods to show that like your product is free of those contaminants. For purity, it's also kind of like very like adjacent to safety too. You want to make sure when you're showing the safety, the efficacy of your product, it's not because some other residues, like maybe like cytokines in the media, like a different kind of cells in the mixture of your cells that is like contributing to the result. That is more general. And then we'll also have identity and potency. For these two, I would say that every cell and every cell therapy are very different. You really want to tailor these assays and these criteria like very like specifically to your product. You can, if you read different literatures about NK cells, like there are a lot of different receptors can be like saying like, oh, these are the best and the markers for mature NK cells, or like these memory NK cells, like these are like different kind of NK cells, but all these things are very good and then like very useful for us to understand our product. But in the end, we needed to test these one by one and to make sure those are understand which are the best representative for what we have. Okay, so those are very like general like guidance about like guidelines we can find online. So if you go online and then look for like FDA guidance, like you will find these too. And you have a lot of papers and then people talking about this. in that very like specific situation, I'm going to do some interaction thing. Okay. So in our company, so like we know we're aware of those guidance, but for us, we need to come from R&D, research and development, all the data like that we have, and then start process development, and then do assay validation, and then do GMP QC. If you're in the field of process development, you understand like assay qualification and an assay like validation is not a small amount of work. it really is a lot. So we want to be very careful about like how we approach process development, and then how many like experiments do we want to have, like do we want to continue from the like R&D into process development. And this is one example. Here's one example. So this is the NK cell profile that I have shown you guys earlier. So our NK cells, this is the when we're doing our R&D, this is what we have established. We test every single marker we can find. Inhibitory receptor, activating receptor, interesting like progenitor receptors, everything we tested. So we got a very like comprehensive package about like what receptors are there on our NK cells. So we have, and this is a good example. So now I'm going to ask the audience, how many questions? How many we have this? If you get to decide, how many receptors should we include in the process development? Let me see if I can see. As many as you can afford. Okay. Thank you so much for considering. Like any other like questions, you can pick a number. You can randomly pick a number too. I'll give it like 10 more seconds. Cool. So let's put it this way. When we do any receptor we pick, we need to make sure we need to do assay validation and qualification for it. If we do any combination of receptors, it means that our like assay qualification to cover single staining and then double staining. If we do 3 colors, we need to cover all different combinations in our assay qualification. So I think technically as many as 40 is correct, and also like as many as like like your group is willing to do. So that for me, like coming from a more like R&D background, this is one thing like I learned like when like moving more and more toward process development, is understand like what need to be done like to finish a good assay, so that we can actually move it more towards the CMC QC assay. Because process development is a gap, it's a bridge between the R&D and then CMC. Eventually, like we want to narrow down to only a couple assays that are critical, that covers covers our safety, covers our purity, covers our identity, covers like potency, like very representative, and then the GMP team can just routinely perform the QC assay as release criteria. Okay. Then here's another example. So we had this very cute assay, like videos, like looking at NK cells killing GMP transfected cancer cells. Raise your hand if you think this is a great, raise your hand if you think this is a great assay that we'll be able to like convey that assay into like process development with no problems. Thank you. One person raised your hand. Pretty good, pretty good. Yeah. Yeah, it should not be any problem, right? You have one set, like you have one single cell line, like there's like like NK cell, you just like put them together. There will definitely be a way to quantify them. Okay. So I guess most of people don't think it's a good idea. Can anyone share a little bit why this is not a good idea? What kind of problems can you already imagine we'll have in process development? Great. Cool. Fantastic. So one of the like a lot of people hit on one of the most important part. It's like this is a qualitative like assay. Like we really want to make sure the release assay is quantitative, so it can be measured into numbers and then like with statistics and then like compare between different batches. It's one important thing. And then to combat that, we can also say that there will still be ways to quantify this. For example, this, we can use play reader to measure the fluorescent intensity, and then we just need to do the proper qualification and then like assay development for it. But the other thing that we don't really, for us, like we learned throughout the process is also that having GIP or with or without GIP is actually a much bigger deal in process development than I thought. So in R&D, we will just, if we need a different color, we just do a transfection and then have a stable cell line, and then we can just have and all the colors we want. But in process development and eventually into GMP, if there's any manipulation we're doing, like in the cell line, even in the cancer cell, we really need to show that they're comparable. So if you want to use, if there's a GMP grade, there's a GMP grade like GMP cells available, that's the best. If there's like nothing available, if you reintroduce fluorescent into the cancer cell yourself, which really means that you need to compare your untransfected cell line and your stable cell line and show them comparable in very different matrices. So it's a very interesting. So these are just two examples showing that the mindset and then from R&D, how our mindset the kind of advance from the R&D setting into a process development setting. Next, I want to quickly share a little bit about, as working on pluripotent stem cell derived NK cells, what kind of challenges did we encounter, specific challenges we encountered. So one thing we have encountered is that because we are doing pluripotent cell derived NK cells, meaning that it needs to go through a relatively long differentiation process. The time is just there, because if we're mimicking the developmental process in our body, it just takes that much time to get to the phase when our body developmentally that we can see it in immune cells. So we're just looking at this very long differentiation process. This is not a problem for research and development, but in process development, and then CMC, that means we need to have proper interval, also proper like monitoring like intervals like in process testing that we will do to make sure the whole manufacturing process on the right track. You will have to pick like the intervals you have to do. Like it can be, ideally it will be like as many as you can, but like in reality that would be very difficult to do, like with how many people we have, and then also how much resources we will need to accommodate that. So we learn and then narrow down to what's ideal for our process. And the other challenge we encountered very specifically for NK cells is the drug delivery like aspect. So we eventually decided to inject NK cells through IV. And it does means that the safety consideration will be higher. So we really want to make sure they're safe. And then there's a lot of testing we have to do for this trajectory. But it will be the best for the cells. And the other interesting like decision we needed to make is whether we would like to cryopreserve our cells or to deliver our NK cells live. Again, another quick question to the audience. Another like question to the audience, like if I ask you to pick between cryopreserved NK cells or delivered NK cells live, what would you like, which one do you think is it's a better option? Cool. Yeah, so like most of the most of you suggested that like cryopreservation will be like most of you like picked cryopreservation with very good reasons. Like for every batch we will generate for CMC, it means that we need to finish like release criteria for that batch. Cryopreserve meaning that the cells can be frozen down, like we can do a relatively big batch and then we just do assays for once and then the whole batch are good. If you do live, you're basically every patient you need to produce some live cells for them every single time. So this is a lot amount of work. So ideally, we would really prefer cryopreservation, but we also need to look at the cells too. So we're still having the based on my like understanding, like based on my own experience in the field about NK cell cryopreservation, we're still trying to have better cryopreservation, like cryopreservation method for NK cells in particular, because once you cryopreserve them and you thaw them, you lose cells, and then the viability can be really low. So this is something we're still learning right now. Cool. And at the very end, I want to also share a little bit more about so like I mentioned, HebeCell is tiny. We have 30 people, but we need to do R&D, process development, and then CMC. So, and then we have a like established process right now. Like we have all the assays set up. Like we are producing a batch in the clean room right now. What's my experience with this? So I would say it's very difficult and very challenging when you have a very small team, when you have to do CMC. So process development and then CMC, you just need a lot of hands to do this. But there are also benefits about like if you're having a small team, if your team is great. is if your team is great, then having a small team also meaning a very much closer communication, much open communication channels between the different departments. And then for a new therapy product, like for someone like us are doing a new cell therapeutic product, having closer communication is really critical for the final success and final product. If I have any like suggestions or recommendation, I would say that like train everyone. It's like oh, like even if your R&D team is not going to be involved with process development and CMC, train them and make them like and help them understand what the end product will look like. So like they can set up the assay with those mindset, which will help the downstream process like significantly. And then that will be it. Thank you so much for your time.
Great. Thank you so much, Dr. Wang, for sharing all that and kind of going through, process development and kind of sharing some tips with us. We do have some questions that came in. So the first question that was asked was, do you use flow cytometry as a quality assay, or what method do you use to QC?
Yes, we do. We do use flow cytometry. I think it's a very like good assay for cell therapy in particular.
Great. The next question that we have is, what are the passing quality criteria and how stringent are they? And how in-depth does each lot need to be tested?
Every lot has to be tested. You do have to test every single lot. If you have sublots, you also have to test every single sublot.
There's no shortcuts.
No. The one thing you will have to do is make sure you have safety tested for every single lot you have, because that's the one thing the FDA cares the most. And then the other part about like what like assays that do we use for every single lot. So assays is really like you want to have assays that hit all the 4 criteria that I mentioned, like safety, purity, potency, and then identity. So you just want to make sure your assays covers all of them, all the different aspects. Which particular assays? I don't think I can recommend anything, because every cell product is different. And then know your cells, and then pick the assay that's they're the most representative for your cells.
I've heard you say know your cells so many times today. I feel like you need a t-shirt that says.
Well, it is the I feel like the most important thing for like doing process development is like you have to know your cells, you have to know your product.
Absolutely, yeah. So our next question is, can you share more about the cytokines you are using to differentiate into NK from CD34?
I think if I answer that question, my CSO will be very, very unhappy about this. So sorry, no.
Okay, it's all good. We'll move on. The next question is, do you have the chemical defined HebeCell cell differentiation protocol? Do you have like a protocol that you can recommend?
There are protocols available online, and then a lot of the papers have good differentiation protocols. But the challenge is really not about like, oh, if you can you get NK cells? Like I believe I haven't tried all of them. I haven't tried all the recipes online, but I think a lot of them you can get some NK cells in the end. But for us, the challenging part is how we can produce a very comparable amount of NK cell every single batch using our like platform, using our process, and that is the most challenging part. And yeah.
Thanks. So the next question is, how much of each lot is used for testing?
So there's one thing that is set. So if you do USP 71, which is the standard sterility testing of your cells, of your product, you have to do 10 to 20% every lot. So 10 to 20% of your lot will have to be like sent out for like USP 71 testing. If you're going to have a backup portion of that, like if you want to have a backup sample, there'll be another 10 or 20% of the whole batch you're producing. So that number is set. For other assays like potency, identity, and other assays, it really depends on your assay. So you just need the amount that you can finish you need for your assay.
Great. So the next question is, how long does it take you to produce NK cells?
We get our NK cells like 30 plus days.
So the next question is asking, have you looked at PBMC to differentiate to NK cells?
Yes, we have. But for us, there are benefits about like using pluripotent stem cells as a starting source. So that's why we went with it. Yeah.
Great, thanks. So there was a question where it says, so you gated your cells on CD56 positive when you showed the quantification of inhibitory and activating receptors. What proportion of cells are CD56 positive?
We get over 95% of CD56 positive cells in the final product.
Okay, great. So the next question is saying, you mentioned your platform uses 3D culture system. How scalable would you say this is, and what are the main advantages over 2D systems?
I think like kind of the second part is the answer to the first question. Like the reason we try so hard for a 3D bioreactor system is because of the benefit of like how scalable it can be. So for us, like looking at this NK cell therapy field, we would maybe someday we would like to see like we can have this established enough. It can be similar to like the protein like modality we have right now. You have like 10-liter like bioreactors you just keep producing. So hopefully we'll get there sometime.
Great. So our next question is, how many markers do you use to confirm NK differentiation, and which markers do you suggest for confirmation?
So for us, we actually did 20, 30, 40 markers during our like research development process. We did a lot of them. We did a lot of batches of the process we wanted to do, and then eventually we narrowed things down to a handful. Which receptors exactly? You will see it when we have a product, but also so it's really it can be really different from like between different products. So if you go online and then you can look, there are some NK cell product, they really looking at like CD16 like percentage. There are other like looking at different like activating like receptor percentage. But in the end, you want the marker to be really representing like the potency and the identity of your cell. So just choose a handful and then which are really, really consistent in your process.
Are there any like common ones that are, generally go-tos?
So I have seen different things. If you go online, like I have seen, like I said, like CD16 is a very popular one. There are all, I think I also see people like, well, CD56, of course. And then I also see like NKG2D before. So like people really use different things, and then just yeah, which are more representative for their products.
Great. Thanks for expanding on that. So the next question is, do you need feeder cells in the process to generate your NKs?
We don't, which is a very unique point for our like product and process.
have you explored direct conversion of PBMCs instead of using iPSCs as starting material? What are the challenges? So I think you had addressed that kind of previously. Do you kind of want to summarize again?
So from my own perspective, we have tried, and then like we, but they both have their like pros and cons. And so in the end, if you have a very robust platform that you can generate either from PBMCs or like um, polypotent stem cells into NK cells, that would be great. So in our hands, we just have like more consistent result from like polypotent polypotent stem cell derived NK cells.
Um, and then one question here, can you use NK cell therapy in conjunction with other types of immunotherapy or other treatments? Would there be any benefit when used in conjunction?
And that's a very good question. The answer is yes. Like NK cell therapy probably will work better with like in the combined therapy with some other the standard like therapeutic like established right now. The benefits are like a lot. So for example, like NK cell itself is a like relatively short-lived cell type in our body. So and that's kind of also related to the benefits, which is they don't like induce very strong like immune responses, but which also means that NK cells are relatively short-lived. So like if they target cancer cells for a while, they will die in in weeks instead of like T cells, they can be there forever. So if we can pair this with other like for example like immuno checkpoints or things like that, like it can really enhance the benefits of NK cell in this like shorter amount of period. It's great. It's a great thing. Like in the ideal world, we will definitely just be like setting up filing an IND and doing a trial like with like NK cells plus something. But in reality, the challenge will then be if you want to design a clinical trials, like a clinical trial that is NK cells plus something, the clinical setup will be multiplied. So that is something that we have to take into consideration when we are deciding what's the final form of the product would be.
Right. We just had one more question come in asking, did you examine how long your 3D generated NK cells can survive after delivering to patients?
So right now we cannot do that because we cannot inject NK cells into patients. But we want to know, so that's why we are like going through this IND filing so we can like start like the phase 1 trials with FDA. But like we do need to do like assays, and then we need to have like qualifications about to address this. Yes, we do studies to address that.
Right. All right. Well, I just wanted to thank everyone who, asked their questions. There were some great questions today. And then, of course, thank you, Dr. Wang, for your wonderful talk. If you guys do have any questions after the webinar is over, I do have a slide at the end that has Dr. Wang's email address, so you can feel free to reach out and ask her any questions that you might have. But with the last few minutes of the webinar, I do just want to share a few product slides with you guys that might be interesting for this group. And I specifically want to highlight our humankind cytokines and growth factors. So as I mentioned in the beginning, we do offer both RUO and GMP grade proteins for NK cell differentiation and maintenance. We have all the proteins necessary for this workflow, such as various interleukins, SCF, BMP4, FGF2, and more. And I also just want to emphasize that we do have the largest GMP protein portfolio on the market. So our humankind GMP products are qualified as raw materials for many research areas, including cell therapy, regenerative medicine, and tissue engineered products. We have a complete GMP cytokine offering for CAR T, macrophage therapy, dendritic cell therapy, and stem cell therapy. Additionally, humankind products are also currently being used in phase 2B and phase 3 trials. So the main advantage of our humankind proteins is that they are authentic human proteins because they are raised in human HEC293 cells. So they have native glycosylation and protein folding. This is often lost when proteins are expressed in other systems. Additionally, we have seen that humankind proteins have superior bioactivity, which means you'll get enhanced results and healthier cultures. Because of our quality manufacturing process, you can also be confident that our proteins will be animal component free, xeno free, endotoxin free, tag free, and carrier free. And then I also just want to highlight kind of the superiority of our thermostable FGF2. So at Proteintech, we understand, the headache of having to continuously replenish FGF2 in your PSC cultures. And this, not only can be time consuming, but it can end up costing quite a bit of money. We actually engineered a thermostable FGF2 for enhanced stability in culture media without having any alterations to its biological function. So when you compare our thermostable FGF2 to the NIBSC standard, you can see that our FGF2 is 12 times more active, and it degrades 3 times slower and can maintain pluripotency even with a 2-day feeding cycle. So overall, with this thermostable FGF2, you're not only getting a product that performs better, but also lasts longer. So ultimately, it saves you time, money, and kind of the headaches of culture integrity. Additionally, because of the advantages of our humankind proteins, we have seen that this translates to better T cell expansion and proliferation. So here on the left-hand side is a comparison of activity of human proteins compared to E. coli and insect-derived proteins. So we compared IL-1 beta, IL-6, IL-23, and TGF-beta-1 and determined Th17 differentiation of human CD4 positive cells. The human proteins are represented in the blue bars here, and you can see that in each instance, the human proteins have superior performance compared to proteins derived from insect and E. coli. On the right-hand side, we have a comparison of our IL-2, which we're showing here in the green bars, versus a competitor IL-2 shown in the red bars. And you can see that our IL-2 resulted in greater levels of gamma delta T cell expansion and proliferation. So as I mentioned earlier, we do have a large GMP offering, and we understand, the importance that quality can have on safety, efficacy, and reproducibility, and ultimately, affect the commercialization of cell and gene therapies. So humankind GMP abides by all relevant guidelines and testing that's listed here, so that we can ensure the highest quality materials for our customers. And all our GMP products have the following documentation. So you can expect a certificate of analysis, certificate of origin, safety data sheet, certificate of compliance, certificate of viral testing, animal-free certificate, certificate of sterility, and data sheet for specifications. And then lastly, we understand the importance of lot-to-lot consistency, especially when you are using GMP quality materials for cell and gene therapies. So here is an example of our PDGF-BB and Activin A, and we observed that between 3 independent lots, the activity and the purity was nearly identical. So this can ensure data reproducibility and quality. So overall, with our humankind GMP products, you have access to the largest GMP protein portfolio that is in stock for quick delivery. You can also be confident in our quality as we are ISO 13485 certified. We ensure quality checks at every step of production. We also have ISO-rated clean rooms for manufacturing and filling. We test the quality of our raw materials. We have a production process to generate animal component-free and xeno-free proteins. And for our customers, we also have quality assurance support and our facility that's available for on-site audits. So if you do have any questions regarding our GMP products, please feel free to email Shrithu Sankar, who's our product manager for Humankind. And you can email him at sreethu@ptglab.com. So with that, I just want to thank everyone for joining the webinar today. If after I end the webinar, you do have any questions, please feel free to email us at events@ptglab.com. Or if you do have any further questions for Dr. Wang, you can email her directly at swang@hebecell.com. And if you like today's event and want to learn more about our upcoming events, please do follow our social media because we do post about all our upcoming webinars on there. And I do just want to mention that we will be sending a follow-up email where you will receive a recording of this event as well as a certificate of attendance. And please do allow 1 week for this follow-up email to be sent. So with that, thank you everyone for joining. Thank you so much, Dr. Wang, again for that wonderful talk and for the really great Q&A session. And I hope everyone has a great rest of their day. Take care.